populate because it does it at different times for different people. Okay, so we'll get started. Hello, everyone. Good morning, good afternoon, good evening, wherever it is that you are. Welcome to the first week in the fall 2020 iteration of the Long-Term Animal Research Seminar Series. We start off this new set of talks with talks from Dr. Sarie van Belle and Dr. Marco Festa-Bianchet. And before I introduce Sarie first, I'll make a couple of announcements. For anyone who's new to the series, I'm Matthew Zippel, and this fall I'm co-organizing the series with Liz Lang, who's also here. The goal of this seminar series is to highlight the power of long-term studies of animals to elucidate otherwise hidden phenomena in animal behavior, life history, population dynamics, and evolution. If you are participating via Zoom, then at the bottom of your screen, or maybe at the top of your screen, depending on your version, you'll see a Q&A tab. If you open that tab, you'll be able to type any questions that you have, as well as see and upvote other people's questions. So then at the end of the talks, we'll go through those questions, starting with the ones with the most votes. However, if you have a clarifying question that you feel like needs to be addressed during the talk in order for you to understand something, you can type clarification in capital letters at the start of your question, and both Sarie and Marco have said that they'll do their best to answer those questions in real time. Lastly, recordings of all of the talks will be available on YouTube shortly after they conclude. So if you need to leave early or know others who are unable to attend live, this talk and all the others will be available for viewing and reviewing after they are complete. So now I'm very pleased to introduce today's first speaker, Dr. Sarie van Belle, postdoctoral research fellow in the Department of Anthropology at the University of Texas at Austin. Sarie received her PhD from the University of Wisconsin and held previous postdoctoral positions at the University of Illinois and at the National Autonomous University of Mexico before arriving at her current position. Sarie studies primate behavioral ecology and sociology, or sociality, sorry, and she does so in a long-term study of black howler monkeys that she established herself in Palenque National Park in southern Mexico. And one thing that's really exciting about her work is the scope of the questions that she's been able to ask, ranging from questions of collective action, social organization, and dynamics within and between groups to fundamental questions of population dynamics with important implications for conservation. So I'm now really excited to welcome Sarie, and I'll turn things over to her now.
Thank you very much for this nice introduction and for I'm sorry. Thank you very much for this nice introduction and for inviting me to be part of this interesting webinar series. Today, I will talk about dispersal regimes in black howler monkeys. Studying dispersal patterns is notoriously difficult as it generally occurs infrequently in an individual's lifetime, or it might be difficult to follow a dispersing individual, especially when it moves outside the study area, or unmarked individuals might not be recognizable outside their original group. And hence, gaining insight into a species' dispersal pattern is often based on long-term demographic data that steadily accumulates data on migration events. Alternatively, dispersing individuals move alleles around, particularly when they reproduce in their new group, and this gene flow influences the genetic population structure. So we can examine that genetic population structure to arrive at the underlying dispersal patterns. And combining both approaches leads to even more insightful revelations. In animals, dispersal regimes are generally considered to be either sex-biased or bisexual, but there is considerable variation in that not all individuals of the dispersing sex might disperse, or some individuals of the philopatric sex might leave their natal group. Even within the bisexual dispersal regime, there might be variation in dispersal rates between the two sexes, or differences in mean dispersal distance, with one sex generally dispersing further than the other. Individuals may also disperse together with other group members or may disperse into groups where a group member had previously dispersed too, referred to as parallel dispersal. Immigrants might not stay forever in their new groups and they can disperse secondarily after they have or have not reproduced in that group. Lastly, dispersing individuals may or may not successfully immigrate into established groups, and the success rates can differ between the sexes. If immigration is not successful, extra group individuals of both sexes can attempt to form a new group. And all these elements can occur in one population, making it difficult to interpret the genetic signals that they leave behind, and thus long-term studies are needed to fully grasp what is going on in terms of dispersal. So today I'm presenting my findings on the dispersal patterns of black howler monkeys based on 15 years of demographic data combined with molecular data. My study takes place in Palenque National Park in southeastern Mexico, and I wouldn't have been able to establish this long-term study without the collaboration with my Mexican colleagues Alejandro Estrada and Alsa Barrios. I have been studying 6 social groups. The project started with a pilot study in 2005, followed with my PhD dissertation in 2006 and 2007. And I returned to the field long-term in 2010. Groups were gradually added, some disintegrated, and new groups were formed. Throughout virtually the entire study period, I have been collecting fecal samples for genetic analysis from all group members. I also conducted 2 surveys to collect fecal samples of additional groups as well as of those newly formed groups. This map shows the relative location of the main study groups and the 13 additional groups surveyed in 2012. Throughout this project, I have been collaborating with Tony Di Fiore to analyze the samples of almost 170 individuals, each for 21 polymorphic microsatellites. To confirm dispersal events, I searched for complete matches of multi-locus genotypes among individuals that initially were thought to be different. Obviously, when 2 individuals residing in different groups turn out to be the same individual, there is evidence of dispersal of that individual. I also used parentage analysis to search for the most likely father and mother of adults. If the mother and father resided in the same group, it was a strong indication that the adult in question was born in that group. If the adult did not reside with its parents, it must have dispersed. So throughout the study, 21 sons were born in the 6 main study groups that survived their first year. 15 of them disappeared from their natal group at an average age of 4.7 years, so they were young adults. Of these 15, I confirmed the dispersal of 8 of them. 7 dispersed into an already established group and 1 formed a new group. The 6 sons that remained in their natal group were still juveniles and hadn't dispersed yet. Not surprisingly, none of them reproduced in their natal group. Looking at daughters, 19 were born in the 6 study groups that survived their first year. 9 of them disappeared from their natal group at an average age of 4 years. But this was largely skewed by 1 daughter who disappeared at an age of 9.5 years. The other daughters were between 3 and 3.5 years old. Of the 9 disappearing daughters, 2 were confirmed to have dispersed, and they did so together and they formed a new group. Of the 10 daughters that remained in their natal group, 2 were still juveniles, and 8 reached adulthood and reproduced in their natal group, as did the daughter who disappeared at a later age. So looking at this in a little bit more detail, this figure shows the home ranges of each study group per study year. So the 8 sons that were confirmed to disperse did so to neighboring groups. Mafu and Kimbo, who were full brothers, dispersed together, and thus dispersed 2 times. The 2 daughters of the Pakal group formed a new group with the son of the Motiepa group, in between their respective natal groups. Now, focusing only on males, Let's add the confirmed dispersal events of adult males, 10 in total. Most dispersed to neighboring groups, but 3 dispersed 2 home ranges away. Both Kriek and Goose, originally from the Balam group, each dispersed together with sons born in their group, yet they did not so with their own son, but with the son of the other male. Jupiler, who was present since the onset of the study as an adult in the Motjepa group, dispersed at an age of at least 17 years old to the neighboring group. And Ugo, who was the other male in the Motjepa group since the onset of the study, together with Jupiler, is the son of Jupiler and a female in that group back then. And he is the only male to be known to reproduce with an immigrant female in what is believed to be his natal group. There were also 9 immigrant males whose origin we don't know, so suggesting that they dispersed from further away. Let's do the same with females, adding the confirmed dispersal events of adult females, only 3 in total. The 2 females, Moshe and Akira from the Palam group, abandoned their group to join the males of their neighboring group. And Pura was an adult female in the Pakal group in 2005, and she disappeared in 2016 at an age of at least 19 years old. But 60 months later, she returned to the study area and established a new group with an extra group, male and female, next to her original group. We also observed 4 immigrations of adult females whose origins we don't know. I also analyzed the spatial genetic population structure and only using data of 2012, including 18 groups. The isolation by distance model states that if dispersal is spatially restricted, then demographic units that are geographically close to each other should be genetically more similar to one another than demographic units further apart. So basically, this model examines the correlation between the geographic distance and the genetic distance between pairs of demographic units, and that shows groups as the demographic units in this model. If dispersal is not spatially restricted, you should not find a correlation. If it is restricted, you should find a positive correlation, and a negative correlation indicates obliged far distance dispersal. I also conducted spatial correlation analysis, which is based on the same principle of examining the correlation between a measure of genetic similarity, and here I used relatedness values between pairs of individuals, and a measure of spatial distance between these 2 individuals, but dividing the spatial scale into separate distance classes. A mental test is then used to verify whether the observed pattern is different from what would be expected if dispersal were to be random. The results are generally shown in these types of graphs, with the yellow line representing the observed pattern and the gray area the random patterns. Higher than random values suggest that at these particular distance classes, there are more pairs of individuals that are closely related to each other than would be expected by chance. And thus in this example, there is a high rate of individuals dispersing to second distance class, with the first class generally representing intra-group diets. So for our results, the isolation by distance analysis revealed that dispersal is spatially restricted for males, but not for females. And for the autocorrelation analysis, I used the number of home range borders between pairs of individuals for the distance classes. Both males and females showed the same pattern in that pairs of individuals were more likely to be related when residing in the same group, but not when residing in different groups, irrespective of how far these pairs of groups were located. So for the discussion, the genetic population structure analysis based on survey data, including 18 groups with the furthest 2 some 2 kilometers apart, suggested that within this spatial scale, male dispersal seemed to be restricted, but not female dispersal, and a wider survey area might have revealed more information. However, by combining long-term demographic and molecular data, I was able to confirm 32 dispersal events of known origin and additionally 13 immigration events of unknown origin. So like other howler monkeys, the black howlers have a bisexual dispersal regime, yet there is a strong bias towards males, with virtually all males dispersing from their natal group, while over half of the females stayed to reproduce in their natal group. There seems further bias in terms of dispersal distances, with males tending to disperse to neighboring groups, while that was less clear for females, although possible. Nonetheless, both male and female immigrants also arrive from groups outside the study area, suggesting longer dispersal distances are possible too. For both males and females, we observed pairs of individuals dispersing together, and in males, we further recorded individuals dispersing into the same group as previous group members had done. We also confirmed secondary dispersal for both males and females, and both after effective dispersal when they had reproduced in that group, and after ineffective dispersal without reproducing. Lastly, both males and females were able to immigrate into established groups, yet more males did so. Whether that is because more males dispersed than females, or more males are successful, is yet not clear, because we rarely observe these immigration events, and the ones we have observed involve aggressive chases for both males and females. Not surprisingly, within a bisexual dispersal system, extra group individuals can successfully form new groups and carve out home ranges among the already established groups. And to conclude, black howlers aren't any different than any other species in that the dispersal regime is very complex, and it takes a while to figure out how complex. And with that, I want to thank you for your attention.
Great. Thanks very much for that talk. I think we have time for a question or two. Maybe I'll get us started. In a bisexually dispersing species, it seems like that presents additional challenges for knowing when animals die, right? If it's sex biased and then that sex, the phylopatric sex disappears, they've probably died. So I guess I'm wondering, how do you assess whether or not an animal is likely to have died? Yeah.
We had a hard time. Well, we have found some skeletons, but they are too far to actually take a sample and then to try to trace back of who that would be. And it would be assumed that if an individual disappears and dies then alone, that it might not necessarily stay in the same home range. So it's so difficult to just figure out if you find a dead body. Of all the disappearing individuals, we just don't know. Of the female, her name is Pura, who disappeared from her group when she was 18 years old. We assumed she was dead. She was 18 years old, but then she returned a year and a half later. So that just showed to us, it's like, do not assume anything. These individuals might live to 30. Well, we don't know that, but so yeah, we don't know if they die or what they do. We just don't know. So yeah.
Okay, great. Well, thanks very much. I think maybe we will leave it there. Thanks for that great talk.
Thank you.
So Marco, if you want to go ahead and pull up your presentation, I think you should be able to if you just hit share screen. While he does that, I will introduce our second speaker today, Dr. Marco Festa-Bianchet, who is a full professor and head of the biology department at the University of Sherbrooke in Quebec. Marco heads a really exciting lab group in which he uses several long-term studies to understand, well, a wide range of questions, but especially the reproductive strategies and population dynamics of large mammals. He and his collaborators monitor populations of bighorn sheep, mountain goats, and eastern gray kangaroos, and also do work with tundra caribou. So a diverse range of large mammals across multiple continents. His research group has contributed fundamental knowledge to our understanding of reproductive strategies, life history, and aging in natural mammal populations. And his work also contributes useful knowledge for wildlife management and biodiversity conservation. The work that his group does is integrative and foundational. And I'm really excited that he's going to be the first kind of keynote speaker in our fall series. So with that, I will welcome Marco, and I will turn things over to him now.
Great. Thanks, Matthew. And thank you very much for this opportunity to talk to you, even though, as Matthew pointed out, it does feel like a bit of talking to a wall with this system. So I am going to try and pass two major messages today. First, and I'm going to spend most of the time looking at maternal reproductive strategy, so female reproductive strategy, of animals that are at least partially capital breeders. By that I mean that the reproductive success this year depends partly on resources that they kept or that they accumulated during the previous year. And my main message is that really those differences in resource accumulation, so difference in reproductive potential, really determines the reproductive success and also the cost of reproduction. Those who are already have a higher reproductive potential tend to have a higher reproductive success, and that, as you will see, also makes it hard to really quantify the costs of reproduction, the fitness costs. I will then switch to males, and there my fundamental message will be a lot simpler, and I'll try to convince you about just how hard it is to measure reproductive strategies in males, because the cost is not on being successfully reproducing, is in trying to reproduce. And I'll end by pointing out one of the applied implication of the long-term work that I've been doing, and talking about something that it's been in my mind for quite a bit over the last few years, and that is the evolutionary consequences of selective hunting. So I'm particularly pleased to be speaking to an audience of people that are interested in long-term studies. And I really appreciated the previous presentation by Sarie, because long-term studies is what I do. And in particular, I do long-term studies on known individuals. I do long-term study because I'm interested in measuring natural variability. I think it's important that we know how much things like reproductive success, survival, growth can vary from one year to another, and what are the causes of that variability. I'm also interested in, because I'm interested in evolutionary ecology, I'm interested in assessing lifetime reproductive success. So that means you have to monitor individuals from birth to death. I think long-term studies are essential to understand population dynamics. Again, partly because we need to have an idea of how things vary when predator pressure, density, resource availability varies. I'm interested in studying evolution in the wild, and there you need, until recently anyway, you needed a pedigree. You get that with long-term studies. And as I will show at the end of my presentation, I'm also interested in using these results to inform the conservation and the management of wild species. So these are the long-term studies that I've been involved with over the years. The line represents the time when the studies were ongoing. I'm happy to report that all 5 are still ongoing. The thick line is the time when I was involved. So I work with kangaroos in Southern Australia, ibex in Italian Alps, bighorn sheep, and mountain goats in the Canadian Rockies. Most of the data you'll see today come from either the kangaroo study or the bighorn sheep study at Round Mountain, but I'll throw in a little bit of results from mountain goats at Courage and bighorn sheep at Sheep River. The basic technique is pretty simple. I catch these animals ideally when they're young, so that it can identify who the mother is, and I put some kind of marking, ear tags or collar, so I can know who they are. And now with molecular techniques, we can also identify who the fathers are. And I hope everybody noticed the color coordination between the mother and osprey mountain goat in the ear tags. So that is the basic technique, but the success of this research really has to do with the excellent graduate students I've had over time. Here are most of the ones that work with either bighorn sheep or mountain goats, and here are the ones that have been involved with the kangaroo study. So I like to acknowledge the contribution of these people to essentially most of, or all, nearly all the data that you will be seeing today. Speaking of data, this comes from a synthesis of long-term studies of ungulates. And what you're seeing is comparing the average and the range, so not the standard deviation, but the range, the worst year and the best year, of a number of long-term studies that monitor marked individual for at least 7 years. And there's two things, and we're comparing survival of adult females before they get into the senescence stage and survival of young of the year. There's two things that should strike you immediately here. Survival of adult females tends to be very high. On average is above 90%, and it tends to be much less variable than juvenile survival. Juvenile survival, you can see the average all over the place. It's probably about 50%, but perhaps more importantly, the range for adult females, a really bad year is when you have about a quarter of the animals that die, and in most years, survival does tend to be in the 85 to 95%. For juveniles, you go anywhere from 0 to almost 90%. The consequence of this is that the reproductive potential or the reproductive value of the mother, it's nearly always much greater than the reproductive value of the offspring. So you would expect a reproductive strategy that favors maternal survival, because in almost no condition is worth for the mother to increase her allocation to the offspring to a point that compromises her own survival. Because that offspring is likely to die anyway, regardless of maternal effort. And so I will present you data suggesting that the best thing to do for a mother is to first look after herself. And I'll also emphasize the importance of environmental variability. And for that, I'll mostly use data from the bighorn sheep population, the bighorn sheep study at a mountain. You can see here two pictures of the trap. And this population, we can catch most individuals, well, nearly all individuals 3 to 5 times each year. You can see on the picture on the right here, this very complex mechanism where there's a stick holding the door open, the stick's attached to a rope. Inside the trap, there's a block of salt, and at the end of the rope, there's usually a graduate student, and he or she is lying down in a bed where he or she has spent the night, and we'll pull the rope and shut the trap. That's how we get repeated captures of individuals. On the graph, you can see changes in population size, and I'll mostly be talking about, for many of the slides today, I'll be talking about things that happened between the early 70s and the late 90s, sort of the green line here. The reason why I'll talk mostly about the early part of the study is that later on, all kinds of other things happened that sort of made it so that the nice density dependence that we're seeing in those early years kind of went out the window. We had increased cougar predation. We took some animals out, then we brought some in. We had issues with inbreeding, so which created a lot of other interesting results, but most of what I'm going to show you now has to do with that early part of the study. So one way of looking at reproductive allocation is to see how much of our availability of resources affects what females, in this case female bighorn sheep, decide to use for themselves and for their lambs. So here is work by Julien Martin, who was interested in how mass accumulation during the summer varies among lactating ewes and their lambs. So like many temperate northern ungulates, these animals gain up to 25–30% of their mass during the summer, and then use it, so lose it during the winter. So if you catch the same animal multiple times over a summer, you can estimate how many kilos of mass that female or the lamb put on during the summer. And if you look at the top graph, this is kilos of mass gain, so the difference in mass between mid-September and early June by lactating females. And you can see there's obviously a lot of year-to-year variation, huge individual differences, but if you look at the fitted line, there is not much of an effect of density. During this period, the number of using number of adult ewes to estimate population density, it went from a little less than 20 to a little over 100. And this is more or less a flat line, which means that when resources are abundant, here at low density, where resources are scarce, here at high density, lactating ewes, so ewes that providing milk for a lamb, are able to find, on average, about 16 kilos of mass to accumulate during the summer. Again, I want to emphasize there's a lot of variability. If you look at the lambs, the lower panel now, you can see that between low and intermediate density, again, a lot of variability. This has to do with difference among mothers, sex, a number of other variables. They gain on average somewhere between about 19 kilos. But as density goes up, so resources are scarce, their mass gain goes down. I interpret this as a maternal conservative strategy that when resources are abundant, they'll provide enough milk for a lamb to gain mass. But when resources are scarce, they first look after themselves, make sure they get those 15, 16 kilos that allow them to survive the winter. And then if there's anything left over, they'll give it to the lamb. Result, lamb mass gain is reduced by about 17% compared to low density. Small lambs are less likely to survive the winter. So the population dynamic consequence of this maternal strategy is that if you compare lamb survival to population density, number of female sheep, you see this substantial drop in survival. And again, not only this is strong density dependence, again, during those years, but really, as you should always do, look at the numbers. We go from about 70% average at low density to less than 20% at high density. This clearly has drastic effect on population dynamics. And during the same time, the same years, if you look at the survival of prime age ewes, you see absolutely no difference. Again, notice how I change the axis label. It's the same years that I showed you before, but while lamb survival went from, yearly variation went from about 85 to 10%, for adult females, in most years, you're looking at survival somewhere between 89 and 100%. Again, suggesting that these animals will ensure that they do not allocate to reproduction in a way that affects their survival. Not only they're very conservative in what they allocate to the lamb, but particularly when you look at the young females, which are exposed to a greater energetic cost of reproduction because they're still growing, then when density goes up, they simply shut off reproduction completely. So in this graph, we are comparing the proportion of 2- and 3-year-olds that were lactating with, again, a measured population density, number of ewes, and 2-year-olds still have about probably 20 to 25% of their own growth to complete. So while they're lactating, they're finding resources. They also have to find resources to ensure that their body growth is completed. 3-year-olds are at about 90-95% of adult mass. So if you look at the green dots, you can see that the low density, a variable proportion of 2-year-olds was reproducing, but almost as soon as density started going up, 2-year-olds just shut off reproduction, and in most years, none reproduced. For the 3-year-olds, the red dot, there was some variability in reproduction at low and intermediate density, but at high density, them too started not reproducing. This clearly has big impacts on population dynamics, because if your first reproducing class of females moves from 2 to 3 to 4 years of age, clearly population growth rate will slow down. So females use a conservative reproductive strategy. That does not mean that there are no fitness costs. And in this population, we were able to detect fitness costs of reproduction without a manipulation. So I'm going to show you a series of graphs that are all going to look more or less like this one. We are comparing reproductive success in year 2, so year T plus 1, with whether the ewe weaned the lamb, yes, in orange, or did not wean a lamb in blue the year before. And I split up the sample between light females, these are the females that weigh less than the average for the population, so less than 71 kilos, and heavy females, the ones that weigh more than 71 kilos. And you can see that for both categories, there is a cost of reproduction, but this cost is much greater for females that are light and therefore have a lower reproductive potential. So if we look at females that weigh below the average, if they had not weaned a lamb this year, the probability of winning a lamb next year was about 85%. If they had weaned a lamb the previous year, the probability of being successful this year dropped to about 55%. So this is a substantial cost. And that cost is transferred to the lamb that is born this year. So there's a cost of having a brother or a sister the year before. For every female, the same sort of result, but the cost of reproduction was much lower. In fact, if you look at the heaviest female, you cannot find any evidence of cost of reproduction. And this is one of the persistent problems with observational studies. You cannot measure the real cost of reproduction because there is difference among individuals. In this case, because we have the mass of each individual, we can account to some extent for differences in individual reproductive potential. So these are characteristics of the females that play a role in the reproductive cost. There is a number of environmental characteristics that also play a role in the cost of reproduction. This looks like a complex slide, but it's trying to show more or less the same principle for two populations of bighorn sheep and one of mountain goats. So if we look at mountain goat data from Courage here on the left, here is mountain goat, just to remind you how beautiful they are. This is work from Steve Cote and Sandra Mel. And we're looking at how reproductive status this year, breeding, not breeding, influences the probability of giving birth next year. Unlike bighorn sheep, mountain goats sometimes skip a year of reproduction. Bighorn sheep, when they start reproducing, they'll produce a lamb essentially 90, 95% of the year. Mountain goats, maybe 1 or 3 or 1 or 4 years they'll take off. So the probability of having a lamb this year, excuse me, a kid this year is greater, the greenish line, if the female had not had a kid the previous year. So similar result to what I showed just now for bighorn sheep. But this difference is much greater at high density. It almost doubles. So as density goes up, as resources become scarce, mountain goats adopt an increasingly conservative reproductive strategy, which in this species involves also taking more years off. Essentially, again, allocating more to maintenance of the mother than to reproduction. Going back to the mountain, and here we're looking at reproductive success of young females, and I mentioned 2-year-olds have to put up the energetic cost of reproduction and growth. And we are looking at the probability that the female will produce a lamb as a 3-year-old according to whether or not she had already reproduced as a 2-year-old. And at low population density, there is essentially no difference. At high density, females that had produced a lamb as a 2-year-old, 4 out of 5 would not breed the following year. So if they produce a lamb at high density as a 2-year-old, when resources are scarce, most of them skipped the following reproductive opportunity. And if you go to the right, this is actually the only evidence that we have of a survival cost of reproduction in adult females. This is another study population of bighorn sheep in the province of Alberta in Canada. And this population was affected in 1985 by an exotic disease, pneumonia, as a disease that's carried by domestic sheep. So what we're looking at this graph is the probability that a 2-year-old will survive to age 5, according to whether she did, orange, or did not, green, reproduced as a 2-year-old. In years when there was no disease, there was no difference, again, suggesting that they're not compromised their survival. The ones that reproduced as 2-year-old during the pneumonia actually suffered an increase in mortality over the next few years. So this was a small sample size, but this is actually a significant difference, suggesting that this unpredictability in the environment can affect the survival of adults, of young adults in this case, according to how much they allocated to reproduction. So reproductive allocation does have a cost. In most years, it does not affect survival. If you add the stress of an exotic disease, well, it had an effect on the survival. So far, I've only showed you short-term costs. And I started out by waxing lyrically about the importance of long-term studies. What are the long-term consequences of reproduction? For example, does reproduction during early life affect how well they do during senescence? And it turns out that it does. And for this, some recent work by Mathieu Duhart. And Mathieu was interested in how survival or reproduction of senescent ewes, and we call senescent ewes the ones aged at least 8 years or older, and we do so because that's the age when survival probability starts declining slightly. whether their performance was affected by what they did in early life. And it turns out that if they produced a lot of lambs in early life, they had a faster reproductive senescence. And you can see that in the left panel here. The pinkish line is the reproductive, the residual reproductive success after adjusting for various environmental variables of females that had produced at least 3 lambs between ages 2 and 7, and you can see a clear senescence in the reproductive success between the ages of 8 and 16, while the ones that produce less than 3 lambs, there's no statistical ratio. This is essentially a flat line. What's even more interesting is that Mathieu was able to show at cost of offspring sex. This is a sexually dimorphic species. Male lambs are bigger at birth, take more milk, they grow faster, they're about 10% heavier at weaning. So the orangish line here is the reproductive success of all ewes that in early life had produced mostly males, and the other 2 lines are for equal sex ratio and more daughters. So not only there is a reproductive senescence that is brought in by effort, reproductive effort during early life, if they produce more lambs, they have a greater senescence. Again, I underline this is reproductive senescence. Once again, is a cost that's being transmitted to lambs that are or are not born later in life. Mathieu found no evidence of reproductive effort during early life on survival. And here it is. If you want to look first on the left, you can see that not only there is no cost in terms of survival, but the trend is actually the opposite. So the pinkish line, females that produce more or at least three or more lambs during early life, they tend to have actually a slightly greater survival during late life than the ones that produce fewer lambs. You can see the clear overall survival senescence. So the proportion of survival declines quickly. And if you do it by lamb sex, again, the exact opposite. The ones that seem to have a slightly better survival, none of these differences are significant, are the ones that produce more lambs and more males. So cost of reproduction, despite the conservative reproductive strategy, both on the short term and on the longer term. One limitation of working with bighorn sheep is because it's such a seasonal environment, they tend to all be reasonably skinny in the spring and reasonably fat in the fall. So I wanted to look at something that provided more variability, both in environmental characteristics and in individual characteristics. And for that, I went to kangaroos. Obviously, the other advantage of kangaroos, other than the fact that they're extremely cute, is that nearly all the reproductive allocation takes part outside the body of the mother. After a 36-day gestation, the young gets into the pouch, and at that point, the female is in much greater control of reproductive allocation, in the sense that it's way easier for a female kangaroo to pop out a young when she doesn't have enough resources and just walk away, than it would be to go through an early pregnancy and termination for a Euterian mammal that presumably will carry some substantial physiological cost and potential risk of disease. So we first looked at kangaroo reproductive fitness cost of reproduction the same way that we done with bighorn sheep. And here's work by Riel-Geleng. And initially, it just compared reproduction this year 2, to reproduction the previous year. And in this slide, we're comparing survival to at least 7 months of age, what we call the large pouch young stage. Essentially, at this point, we can see the head sticking out of the pouch, and that is when reproduction really starts to become costly, because the young weighs over 1 kilo, so it's quite a bit of weight to carry around for an average maternal weight of somewhere between 24 and 28 kilos, and milk intake also increases. So we expected that females that had weaned the young the year before would have a lower reproductive success this year. The females that did not wean a young, not only there's no significant difference, the non-significant trend is the opposite. So again, this goes back to this idea that I was mentioning, that there's a lot of individual heterogeneity. So some females just do better year after year, not because there is no cost of reproduction, but because they've been able to find enough resources that they can pay the energetic cost of reproduction this year and still have enough left to reproduce again the following year. Now, the way around that is to do an experiment. And this is one of the rare cases where reproductive allocation in a large mammal has been manipulated. And we took advantage of an ongoing study that was interested in using contraceptive to reduce population size of urban kangaroos, to essentially look at what is the reproductive success, again, survival to large pouch young, for females that were not manipulated, and manipulated females, meaning females that received the contraceptive, so that for 1 year, they could not reproduce, and so any resources they got, they allocated to their own growth and maintenance. And here you can see there's a significant difference on the direction that you would expect. So there is a cost of reproduction in kangaroos. It's hidden partially by the fact that kangaroos tend to reproduce when they can afford to do so. And the ones that have more resources can afford to do so year after year. To get more into details of how resource availability affects cost of reproduction, reproductive allocation, we looked at how reproductive success and reproductive allocation vary with changes in body mass, something that you can do when you can catch all these animals at least once a year and look at how their body mass and so the stored resources vary from 1 year to year. And here is work by Pauline Toney. And what Pauline did was looked at the probability of producing a young in year 2 according to the mass change of the mother. So the black line here that shows 0 means a female that weighs exactly the same this year as she weighed last year. Positive values mean female that have gained some mass. Negative value mean females that have lost some mass compared to the previous year. And just to give you an idea, 2 kilos is about 8% of the average female mass. So it's not a huge proportion. Again, I'm used to work with animals that go up and down by 30%, but it's reasonably substantial. So if you first look at females that gained at least say 1 kilo, 4% of their body mass, you can see all these lines are flat lines at 100%. And what they represent are females that had different levels of reproductive allocation the previous year, none, and then increasing length of life of the young, so increasing reproduction to, increasing allocation to milk to reproduction. As females approach this black line of no change in mass, you can see there starts to be a slight decline. And if you go on the left side, females that have lost mass from the previous year, they're much less likely to reproduce. And notice how there's this very sharp decline in these two sort of magenta and bluish line. These are females that the previous year had brought their young to either 7 months or, past 7 months or to weaning. So these are females that made a substantial allocation to reproduction the previous year. And if they lose just a little bit of mass, they're much less likely to reproduce again the following year. So again, evidence of a conservative reproductive strategy that is driven in part by resource allocation, by resource availability. If they can find resources to maintain their mass, then they go ahead and reproduce again. Now, obviously, resource availability is going to depend on resource production. And in the Australian environment, the amount of rain changes a lot, which means the amount of grass availability changes a lot. And the other thing that Pauline did was compare the yearly mass change of female kangaroos, so much change in mass from 1 year to another, according to reproductive allocation. And what she found is that for most females, there were no huge effects of forage availability over, which you can see is a fairly substantial range from 500 to, almost tripled the amount of forage available. But for females who had been successful in weaning their young, then the amount of mass that they lost was substantially affected by forage availability. If there were lots of forage in the study area, they more or less managed to maintain their mass, which allowed them to reproduce again. But in years when forage was less available, they lost a substantial amount of mass, and that led to a decreasing probability they would be able to wean young 2 years in a row. And this really appears to be a conservative reproductive strategy driven by individual condition resource availability. It really looks like at some point the female just senses, I don't have enough resources to reproduce this year. the young gets ejected from the pouch and she just walks away. Obviously, it's extremely rare that we witness this, but we have seen it a few times, and it's not the funnest thing to do in the field. You can see the young is calling desperately for a mother, and she just walks away. And usually this happens in the Australian winter. Within a few hours, the young will not be able, they can thermoregulate very well, and it just not be able to crawl back into the pouch, even if she went back to look for him. Obviously, in this kind of environment, something that weighs only a few hundred grams disappears very, very quickly. There is ravens and other scavengers, but occasionally we have seen this, and it does look like it's pretty much a strategy driven by resource availability. So to conclude, what I'm trying to sell you in terms of female reproductive strategy in these large heteroporous mammals, the reproductive strategy is selfish, and that is because mothers have a much higher reproductive value than their offspring, and any cost of reproduction is moved on to the offspring. And obviously that reproductive allocation depends on resource availability. If there's lots of resources, well, they look after themselves, and then they'll have enough to allocate to the male. So, excuse me, to the young. Right, whoops, what have I done here? Let's switch to males. So here's a large bighorn sheep ram. The most observant about you might notice something funny about his horn. Somebody took an illegal shot at it and hit him in the horns. And that introduces the next subject. I want to talk about male reproductive success and the effects of selective hunting. Male reproductive success is extremely difficult to measure. For females, it's relatively easy. I got a female sheep, she has a lamb, it's her lamb, done. I can compare it to her weight, her age, etc. For a male, I need to sample all the young, use genetic techniques to figure out what the father is, and still, in some cases, I don't know what the father is, because some rams come in during the rut, which I haven't sampled. Some of the resident rams may leave and go rut somewhere else. I know absolutely nothing about their reproductive success. Female, I just need to see the lamb at birth. Okay, she's got one. It may die. I still know that it died. If I don't collect a genetic sample from the lamb, I'll never know what the father is. So I'm showing you a number of caveats about how much more difficult it is to measure male reproductive success, and I've really only started the list. Given those caveats, what do we know about cost of reproduction in males? And one point that I really want to stress is that obviously male reproductive success in a species like this depends on beating up on other males. So the real cost is in trying to reproduce, not necessarily in being successful at reproducing. A large dominant male will serially defend estrus use, and everybody else is scared of him. It doesn't expend that much energy. It's more the younger male that have to do a lot of fighting, running, and chasing that may expend more energy. That being said, I'm going to talk to you about some very recent work by Jan-Erik Stott, who looked at what are the consequences of early reproduction for survival and lifetime reproductive success in bighorn rams. So here, we're first looking at all rams in the Ram Mountain study area. It is lifetime reproductive success, it is age at death, and for this graph, Yanni took out the ones that were shot by hunters, so it's natural mortality, compared to the age of first reproduction. Again, I have to point out, this is the age at which we know that these animals were able to sire a lamb. It doesn't tell us much how hard they try. And if you look at lifetime reproductive success, it's a flat line. If you look at age of first reproduction, it looks like if you reproduce late, you have an increasing life expectancy. Clearly, these data are biased. You cannot reproduce as an 8-year-old for the first time unless you survive at least to 8 years of age. So there's a bias in this data. We can reduce that bias by only considering rams that did first reproduce at 2, 3, or 4, and also that survived to 4 years of age. If you do that, Yannick found that there seems to be a slight increase in lifetime reproductive success for the ones that do not reproduce until they're 4, and there definitely does seem to be an increase in life expectancy. So there seems to be some long-term cost of maybe getting involved with the rut at an earlier age, which does reduce the life expectancy of these males. And I show this because this is one of the few evidence I know of a natural cost of reproduction for large mammal males. I wouldn't want you to think that this is what we find all the time. This is actually kind of an exceptional result. Now, what drives reproductive success in these animals? And in particular, what is the impact of these very large horns, which are the target of trophy hunters? Here's some work initially by Dave Coltman and later confirmed by Alexandre Martin, who looked at how does horn size, or relative horn length here, affect mating success when you account for age of the ram. And what you can see is that horn size has a strong positive effect on mating success for rams that survive to at least 7 years of age, sort of like this one. If they're young, say 2 to 5 or 6 years of age, horn size doesn't play much of a role. They do get some siring success, as I showed previously, but it doesn't seem to depend much on horn size. And this is what you could expect in a system where a mature, say a 7 or 8 year old ram dominant defends the estrus ewe. He's got about 60% probability of being the father of a lamb. Everybody else uses an alternative strategy of essentially trying to cut off the female and mate with her on the run. And that's how they get this reproductive success using alternative mating strategies. Now, bring into this situation hunting regulation that set a minimum amount of curl for a ram that can be legally shot. So this is the definition used in the province of Alberta. Other definitions that are used in most Canadian, until recently American jurisdiction, which means if you buy a license for a trophy sheep, you cannot kill a ram that's got small horns. The one you see in the picture became legal as a 40-year-old, which is the youngest age at which they become legal, and he was promptly shot. Another way of looking at this is that if the horn stopped about here, it would have been illegal for that hunter to kill this ram. So if your horns grow quickly, you become available to the hunt when you're 4 or 5. But those large horns will not help you achieve high mating success unless you survive to, well, minimal to rut as a 7-year-old. So you have a negative selection from the hunt that comes in 1 to 3 years earlier than the positive sexual selection of large horns. This is a textbook case of onset of evolutionary selective pressure. The one that comes in earlier is going to have a greater effect. And I like to point out just how strong this selection is. We very rarely have data on what is the harvest of legal rams. At Ram Mountain, it was about 40%. That means that if a ram was legal to hunt at age 4, it had about an 8% chance of surviving to rut at age 7. Compare that to a ram that is not legal until age 8, he has a 60% chance of surviving to rut at age 7. My point here is that the level of selective pressure that was put upon the sheep by the trophy hunt was very similar to what you might expect in a domestic animal context where farmers select purposely for characteristics that they want. And to illustrate how effective that is, here is at the National Museum of Australia. On the left is the skeleton of a merino sheep when they were first introduced to Alberta about 50 to 60 generations ago. And on the right, the other half of the skeleton is what merino sheep in Australia look like now. You can have some pretty dramatic results with directional selection. And sure enough, Gabriel Pigeon confirmed earlier results by Dave Coltman that over multiple generations of very intense artificial selection, you get an evolutionary change in horn size. So what you have here from Gabriel's paper in blue is the phenotypic change. This was mostly a response to density dependence. And in orange, the genetic effect. So this is the decline, essentially in breeding value of the horns. And so the conclusion, the result here is that during these about 2.5, 3 generations of bighorn sheep, horn size decline genetically. So the evolutionary impact was about 2 to 2.5 centimeters. Not very much, but enough to make the difference between you get shot before you can breed or you survive to breed. The dotted line here shows here 1996 when regulations were changed so that the hunting pressure became a lot weaker, and you can see that the decline stopped. It didn't quite, population was smaller, sample size was smaller. This is not a significant increase after most of the evolutionary pressure was removed, but it's a way of showing experimentally that when you take away the negative selection pressure, selective pressure, the decline more or less stops. Now, I was not, and I still am not, desperately popular with some hunting group because of this result, partly because the result has been abused by other groups trying to say, well, this happens all the time everywhere, which is certainly not the case. But I want to switch gear a bit and show you how you can apply this result to a management context. And obviously, the first reaction would have been, well, this only happens when you study over a mountain, which is isolated, it's got limited gene flow. Do we see this kind of hunting-induced evolution anywhere else? Now, there is no other hunter population with a pedigree, so the next best thing is to look at harvest data. If the size of the horns of rams that have been taken by hunter changed over time. Now, these are very useful because they've been collected for decades, but you have to keep into account some biases. First of all, if the horns are small, the rams cannot be shot. So small horn rams do not show up on your database. And if the horns grow quickly, inevitably that ram is exposed to the hunt younger, so you would expect an age bias. Rapidly growing rams tend to be harvested at a younger age. Once you keep these biases into account and keeping in mind these biases, you find that in areas with intensive sort of quota-free selective hunt, you do see a decline in horn size. So for example, here is 6-year-old rams in Alberta, declined by about 2.5 centimeters over the last 35 years. And the age at which these rams are getting shot has gone up over time. You can look here on the left panel, the average age of harvest went from about 6.7 to maybe 7.4 years of age over these 36 years. And you may say, great, that's what we want. We want to shoot older rams. Well, unfortunately, this increase in age is entirely driven by the proportion of foreign that are in the harvest. Remember, these are rams that are legal. They have large enough horns to be shot, but they have no hope of being a dominant ram. So this is really the case where large horns do not mean high reproductive success. Large horns means you get shot before you can breed. And I draw your attention to a few years here in the 1980s and 1990s, when a third of the harvest was, or close to a third, was made up of rams age 4 and 5. If you look at the more recent years, it's less than 10%. In fact, this trend has continued. And the way I interpret these 4 and 5-year-olds are simply no longer legal because the horns are growing more slowly. Now, there's clearly a problem with this data because I don't have a control. I mean, this decline pattern over time could have been due to some external force. Climate change comes to mind. Luckily for me, the people that do not believe in evolution also don't believe in climate change, but the fact remains that the regulations were similar everywhere. Be nice to have a control. To find a situation where hunting pressure was different, Mathieu Douard went and looked at stone sheep. And in collaboration with wildlife biologists from the province of British Columbia, he identified 2 large populations where the access for hunters were different. In the peace, hunt access is difficult, but in the Skeena, it is really, really difficult. You're looking at days on a horseback. It's hard to get in. The area where hunting pressure was higher, over these 30 years of data, you can see a decline in the horn growth of young rams. Again, this is growth during years 2 and 3. One advantage of these animals is that you can measure annuli on the horns. So even though that ram may get shot when he's 8, you can still measure how much he grew when he was 2 or when he was 3. In the area with lower hunting pressure, there's no overall decline. There's other interesting things going on here. You can see this 10-year cycle, but I don't have the time to talk about it now. My point is that high hunting pressure, you get a decline. Moderate hunting pressure, you don't, which means that you can do evolutionarily sustainable trophy hunting You just have to be careful in terms of the amount of harvest. So this is almost as good as having a control and an experimental population. Now, I'm sure I'm talking to an audience of mostly evolutionary biologists, and at this point, you're probably bored because you're saying, well, why is this surprising? Who could possibly expect something different? You kill the big ones, you let the little one breed, you get smaller sheep. Well, it turns out that this is actually quite controversial, and I can exemplify by the fact that there was an entire special issue in the Journal of Wildlife Management that was dedicated to casting doubt on the idea that intense selective hunting can cause evolutionary change. And one of the centerpieces of this special section was this model, which showed, as you can see here on the graph, that yes, horn size may decline with intense selective hunting, where you're taking 25%, 50%, 75% in all of the trophy rams, but you're looking at the decline here of a few centimeters or 100 sheep generations. 100 sheep generations, about 700 years. So that's not really something that is relevant to current management. So the model was praised by the editor of the Journal of Management, showing that clearly this is not an issue. The more observant among you may have noticed that when you look at a few generations here with a high hunting pressure, there is a bit of a few centimeters decline, but more on that later. There are several problems with this model, all of which contributed to this conclusion. The main one was that they just made up values for additive and phenotypic variance. And this is interesting because there are published values for the additive and phenotypic variance and for the level of heritability of bighorn sheep horns. Now, I wrote a rebuttal, which was replied to by a letter from some of the authors and the editors. And in this one, they used values of additive and variance, phenotypic variance, and heritability that were actually published for bighorn sheep. Now, you may notice some differences between these two graphs. This was the original paper. This was the rebuttal to my letter. For reasons that I'm sure were entirely coincidental, you may notice that the x-axis here is from 50 to 70, while here is from 0 to 70. One accidental consequence of this change in the axis is that while the slope has actually doubled, now that they're using real values, so you go from a decline of about 6 centimeter to one of 14 centimeter, it actually looks the same. You may also wonder what happened to the model prediction for harvest of more than 25% of legal rams, because you can see in the original publication, we have a prediction for 25, 50, 75, 100%. Here we only have 25%. Well, possibly the reason why you don't see 50, 75, and 100% is because if you do apply the model with values that have been published for bighorn sheep, and now let's look at only 10 generations instead of 100 to stay within something that is more relevant to management. This is the 25%. The gray line here is what we actually found empirically at Round Mountain. And this is with 50% harvest, this is with 75% harvest. In other words, a model that was praised in the Journal of Wildlife Management for showing that selective hunting cannot cause evolution, once you plug in data that are available for bighorn sheep, actually predicts pretty much what we found on a mountain, which is kind of satisfying. You can see this is referred to as unpublished because the journal refusing to publish it, but I look forward to the day when the same editor will again praise the model for showing that, yeah, selective hunting can lead to evolutionary change. So I'm done. My take-home message for females is that they're selfish, and they're selfish because mothers have a much higher reproductive value than their offspring. Female reproductive success is limited by resources, so the degree of selfishness will vary with resource availability. Male reproductive success is limited by other males, which makes it much harder to measure. It is based on competition with other males, and so there, I suspect that what we'll find is the real cause, what causes greater mortality, loss of condition, is in trying to reproduce, not necessarily in succeeding. And I concluded by pointing out that in some species, weapon size affects male reproductive success. Weapon size is also what determines the trophy, and trophy hunting can lead to an evolution effect. We can easily avoid it. This is not rocket science. All you have to do is reduce the harvest rate, but it definitely should not be ignored. So I'm glad that these mostly fundamental studies did make a contribution to wildlife management. I conclude by thanking all the students that worked in the different projects and the collaborators that were really the source of most of the ideas that I presented to you today. And of course, we can never forget the funders. I would really like to acknowledge NSERC, the Natural Science and Engineering Research Council of Canada, which is really set up to promote long-term studies. And maybe that's why we have so many in Canada. And thank you for your attention. I'll be glad to take any questions if there are any.
Great, thank you very much. That was terrific, a real tour de force. I have a lot of questions, but I won't make everyone listen to a lot of my questions. Well, let's start with, but we'll try to get through a few of people's questions. And then Sarie, if you stick around at the end, we'll come back to you because Susan Alberts asks a question for you too. So to start, Marco, Susan Alberts asks, well, she says, really interesting results on a wonderful range of species. Regarding those male bighorn sheep, do you know what it is about early life reproduction that's costly for males later?
Okay, the honest answer is no, but because as a professor, I'm a professor, I'll speculate. It has to do with the energetic cost. It could even be direct mortality. These animals occasionally will push each other off a cliff, especially if there's ice around. It was a surprise. I wasn't expecting to see that result, especially given that it does not seem to be immediate mortality. But I think it would happen with some kind of depletional condition during the rut, and then it could even be increased predation during the winter. One limitation of the sheep study, we're only there about 4 and a half months a year. So what happens during the winter is a black box, and you very rarely know what the animals died of, except for the ones that get shot or a few killed by cougars.
And then I had a couple of questions about long-term effects of resource availability. So first in the sheep, whether those lambs that are born in really high density environments but survive anyways, whether there's long-term consequences for reproduction and survival in adulthood.
Yes. First of all, the other limitation there is that those years there was such a high mortality that my sample size of lambs that were born at very high density and then we were able to follow through their lifetime is fairly limited. But graduate student or myself of Fanny Peltier have found substantial evidence of exactly what you're asking. Increased senescence, and this for both sheep and mountain goat, faster senescence in reproduction, and much later age of first reproduction. That was one of the reasons why I stopped. I mentioned I stopped the years where I looked at age of first reproduction during that initial part of the study, because in later years, even though density dropped, very few females reproduced before they were 4, in some cases 5. So yes, there were definitely some long-term effects, and that's one of the reasons why I really wanted to work with kangaroos, where those long-term effects do not seem to be quite as important.
Let's see. Amial Ilani asks, is there a common age for female kangaroos to eject their offspring? So is there perhaps like a limit at which point they no longer do so?
Yes, it seems to be mostly before they get to be about 5 or 6 months of age. So before they get to a weight where they're probably costing a lot to the mother in terms of milk production. And it does look like if they make it past what I call the large pouch, past 7 months, we really have no evidence. I mean, there's still some mortality, but we have no evidence of ejection. Of course, if they make it to that age, then you're mostly looking at the sample of mothers who have not lost mass. Mm-hmm. And the other thing is obviously, you know, when the young is very small, we cannot see it in the pouch, and also we're not there year round. So yes, there seems to be a a bit of a trade-off in that, I shouldn't say a trade-off, but it does seem to be mostly an early decision. Like they ejected what, in my view, is before it's causing a problem in terms of their own mass gain and survival.
I also had a question about the ejection, which is, and I may have misunderstood, but I thought you said that the ejection strategy is potentially less costly than pregnancy termination. And I was curious, I was hoping you might expand on that, because it would seem to me that, you know, females can at least reabsorb some of the energy that they have invested in a fetus if they terminate. But it seems like they lose all of that energy if they would just walk away from an offspring. So I'm just curious what you think the balance of those things is.
Okay, another question that I should say, honest answer is I don't know. I'm, you know, mostly think of the animals I work with, and with ungulates, there is really very, very little evidence of resorption.
Okay.
Female gets pregnant, carries it to term. There is certainly some data with caribou where they collect, you know, in the bad old days where the sampling technique was you go out and shoot 100 and see what you can find. And when they did it soon after the rut, they would find a higher frequency of pregnancy than just before birth. And with caribou, this resorption does happen. I never totally understood physiologically how the resorption works. And in most species, when the pregnancy is terminated, it's a miscarriage. And I'm assuming that a miscarriage involves a much greater physiological cost than just popping the young out of the pouch. I'm not a physiologist, so I don't know. So I hope I didn't say something completely out of, disconnected with reality. But I guess that's just my impression.
Sure, that makes sense.
You know, a miscarriage is costlier than just popping the young out of the pouch. And if we look at, bighorn sheep after they reproduce the first time, is about a 90 to 95% pregnancy rate every year. So it just doesn't seem to be a strategy. I mean, the other strategy that I'm surprised they don't use when they're in bad shape, why don't they walk away from it after they give them birth? And well, maybe animal have been strongly selected that if you give them birth to something, you shouldn't walk away from it, but you should give it milk. So a lot of open questions.
Great. Well, maybe that's the place to leave it with a lot of open questions. This was a really terrific talk. Thanks very much. And Sarie, let's follow up with just one more question for you from Susan Alberts. She asks, how flexible do you think the animals are with respect to the fragmentation of the landscape that you've previously written about? And how much more fragmentation can the population tolerate, do you think, and still be able to disperse?
There was a PhD student who recently finished her PhD. Her name is Karen Klaas, who actually did a study on the fragmented landscape surround Palenque National Park. She didn't get around just yet to do the genetic analysis to really look at dispersal patterns, looking at these two types of models, like the isolation by distance models or the autocorrelation one, but purely on demographic data, it does look like that groups in fragmented areas have less males. And it's not because there are more groups within these fragments. It really truly does look like that males seem to die easier in these fragmented areas. And given that this is the sex that disperse most frequently, it does look like that there is a cost to dispersal more so. We don't know the cost of dispersal in a continuous landscape. There most probably is a cost, but there seems to be a higher cost in the fragmented landscape. And so to what extent can they tolerate fragmentation? Well, if they can't reach that nearby fragment and they can't survive in the fragment they are in because they are born there or because of limit of resources, I think that is what the limits is, how far apart these fragments are and if they can physically get there. These animals get attacked by dogs while they walk on the ground and by other animals as well. So, yeah.
Thanks very much. I really like the thread that runs through both of your research, that these things have important, that the fundamental science we do has important implications for conservation and wildlife management, and that's not something that exists in all long-term systems. So I think that's a nice thread to tie between these two talks. So Sarie, Marco, thanks very much for starting us off with two great talks.
Thanks for having us.
Right, and everyone else, we will see you same time and place next